Hello everyone. The topic of this presentation is the urea cycle, one of the most important metabolic pathways in the human body. We are going to go through this topic step by step, clearly and simply. I am Brinda Ronika, tutor working at Triple M College of Nursing. By the end of this section, we'll be able to define what the urea cycle is, enumerate its functions and purpose, identify the site where the urea cycle takes place, explain each step of the cycle along with the enzymes involved, and understand how the urea cycle integrates with the TCA cycle. The urea cycle, also known as the ornithine cycle or Krebs-Henselt cycle, is a primary metabolic pathway used by humans and other mammals to detoxify and remove excess nitrogen from the body. When we consume protein, the body breaks them down into amino acids. These amino acids are further break down, releasing nitrogen in the form of ammonia. Ammonia is highly toxic. Even small amounts can severely damage the nervous system and the brain. To handle this, the liver steps in and convert this toxic ammonia into a safe water-soluble compound called urea, which is then safely excreted through the kidneys in urine. The entire process is what we call the urea cycle, which is first described by Hans Krebs and Hert Henselt in 1932. So, definition. The urea cycle is a vital metabolic pathway in the liver that converts highly toxic ammonia, a byproduct of protein metabolism, into non-toxic urea. This urea is then transported via bloodstream to the kidneys and excrete in urine, preventing harmful ammonia accumulation in the body. To put it simplify, so here input is ammonia and what process is happening means where the process is happening means urea cycle in the liver, and what output we are getting means urea non-toxic, which is excreted in the urine. The liver takes something dangerous and converts it into something harmless. That is the core purpose of the urea cycle. So now let's see functions and purpose of the urea cycle. There are three major functions are there. First one is ammonia detoxification. The primary function is to convert highly toxic ammonia, a byproduct of protein and amino acid breakdown, into urea. Without this, ammonia accumulates in the blood, causing a condition called hyperammonia, which leads to brain damage and it can be fatal. The second function is safe excretion. So, ammonia is harmful to the central nervous system even in very small concentration. Urea, on the other hand, is non-toxic, highly water-soluble, and can be safely carried in the bloodstream to the kidneys, where it is excreted as urine. The urea cycle makes nitrogen safe for transport and excretion. And the third function is nitrogen balance. The urea cycle accounts for approximately 80 to 90 percentage of all nitrogen excreted by the body. It is the dominant route of nitrogen removal. Without a properly functioning urea cycle, the body cannot maintain nitrogen balance. So these are all the very important three purpose of the urea cycle. So first one is ammonia detoxification, second one is safe excretion, and third one is nitrogen balance. Next we'll see about site where it is happening. So the organ site is liver. The liver, the urea cycle exclusively occurs in the liver. This is why patients with severe liver failure develop hepatic encephalopathy. The liver can no longer detoxify ammonia, so it builds up in the blood and affects the brain. And the cellular site is mitochondria and cytosol. This is a unique feature of the urea cycle. It takes place in both compartments of the liver cell. So the step 1 and 2 occurs in the mitochondria and step 3, 4 and 5 occurs in the cytosol. So this is a key exam point. The urea cycle span two cellular compartments. One is mitochondria and another one is cytosol. Now we are going to see the most important 5 steps of the urea cycle. Each step involves a specific enzyme and each enzyme converts one molecule into another. Let us go through them one by one carefully and clearly. The step 1 is formation of carbamyl phosphate. So formation of carbamyl phosphate which is occur in the mitochondria. This is the first and the rate limiting step of the urea cycle, meaning this is the slowest and most controlled step. Here ammonia ions combine with carbon dioxide to form a molecule called carbamyl phosphate. The enzyme responsible for this reaction is carbamyl phosphate synthase 1, which is located in the mitochondria. Here key points to remember is the step consume 2 ATP molecules, it is energy expensive. The reaction is irreversible. Once it happens, it cannot go back. CPS1 requires a cofactor called N-acetyl glutamate for its activity. So NAG acts as an allosteric activator. There is also a second enzyme called CPS2, but that one is found in the cytosol and it is used for pyrimidine synthesis, not the urea cycle. So don't confuse the two. So in the summary, ammonia plus carbon dioxide which formates carbamyl phosphate by CPS1 and needs 2 ATP and NAG in mitochondria. Next, step 2, formation of citrulline. It occurs in mitochondria. In this step, carbamyl phosphate reacts with ornithine to form a new molecule called citrulline. The enzyme here is ornithine transcarbamylase, OTC. So now remember ornithine, it is a carrier molecule of the urea cycle. It enters the urea cycle, picks up chemical group and eventually gets generated at the end, just like a recycling system. After citrulline is formed in the mitochondria, it needs to move into the cytoplasm. This is done by the transporter protein called ornithine translase. So to remember this step, carbamyl phosphate plus ornithine which forms the citrulline. Citrulline moves from mitochondria to cytoplasm. So from step 3 onwards, everything happen in the cytosol. So first 2 steps which is occurring in the mitochondria, and from 3 onwards cytosol. So now we'll see the step 3, that is synthesis of arginosuccinate. It is occur in the cytosol. Now we are in the cytoplasm. Here citrulline reacts with aspartate to form arginosuccinate. The enzyme is arginosuccinate synthetase and this reaction requires ATP. So here is a very important point. Aspartate is the source of the second nitrogen atom that will eventually end up in urea. The first nitrogen came from ammonia in step 1. Where does aspartate come from? Aspartate is formed by transamination of the oxaloacetate and glutamate via the enzyme aspartate transaminase, and this reaction requires vitamin B6. So this is clinically significant when we check liver function test. We measure AST levels now in the blood. So if AST level is elevated, it means liver cells are damaged and leaking this enzyme, which is also affect the urea cycle. So here citrulline plus aspartate plus ATP, which forms the arginosuccinate. So next we'll move into the step 4, that is cleavage of arginosuccinate. So it occurs in the cytosol. So in this step, arginosuccinate is split into 2 products. One is arginine, which continues in the urea cycle, and the second one is fumarate, which is released as a byproduct. So the enzyme is responsible here is arginosuccinate lyase. So now fumarate is very important. It is not just wasted. Fumarate enters the TCA cycle, Krebs cycle in the mitochondria, where it contributes to the generation of NADH. So it is also involved in the trisoine catabolism. So this is connection between the urea cycle and TCA cycle. So next, step 5, which is formation of urea. So which is also occur in cytosol. So this is a final step and the most satisfying one. This is where urea is actually produced. So arginine undergoes hydrolysis. So hydrolysis means addition of water by the enzyme called arginase to produce urea. So urea which is released into the blood and transported to the kidney for excretion. And second one is ornithine, which is regenerated and transported back into the mitochondria to begin the cycle again. So this is regeneration of ornithine is what makes it a true cycle. Ornithine is both used and recreated in every round. So the urea produced here travel to the kidney, gets filtrated and is excreted in urine. That is how the body gets rid of its nitrogen waste. So arginine plus H2O2, so we'll get urea plus ornithine. So then again this step, it will start from step 2. As you can see on this slide, here is a complete diagram of the urea cycle and the 5 steps of urea cycle. The step 1 is formation of carbamyl phosphate and the step 2 is formation of citrulline and the step 3 is synthesis of arginosuccinate and the step 4 is cleavage of arginosuccinate and the step 5 is formation of urea. Take a moment to trace the pathway from the carbamyl phosphate formation in the mitochondria to the release of urea in the cytosol and the regeneration of ornithine to begin the urea cycle again. So please make sure you can draw and label this diagram in the examination. So next we'll see about the integration with the TCA cycle. Now let us talk about one of the most exciting parts of the cycle and in biochemistry, the integration between the urea and TCA cycle. This is also called the Krebs bicycle because both cycles are connected like two wheels. So the first integration is the link, the fumarate. The urea cycle produces fumarate in step 4. This fumarate enters the TCA cycle in the mitochondria where it becomes malate then oxaloacetate. And the second one is aspartate recycling. So that oxaloacetate is then converted back into aspartate via transamination and that aspartate re-enters the urea cycle in step 3 bringing the second nitrogen atom to make urea. And the third integration is energy balance. The urea cycle consumes 4 ATP molecules per cycle, but the TCA cycle generate about 12 ATP. So giving a net energy gain. So overall the body is still gaining energy. And the fourth one is CO2 supply. The CO2 released by the TCA cycle in the mitochondria is used as a raw material for step 1 of the urea cycle. So that is a formation of carbamyl phosphate. The two cycles literally feed each other. And the fifth integration is metabolic flexibility. So the oxaloacetate produced can either re-enter the TCA cycle or be used for gluconeogenesis making glucose. So this shows how these cycles are connected to the entire metabolism of the body. Now let us briefly look at the clinical side metabolic disorders of the urea cycle. Defects have been reported each of the 5 enzymes of the urea cycle. All of these disorders leading to buildup of ammonia in the blood, a condition called hyperammonia which is highly toxic. The clinical symptoms associated with urea cycle enzymes defects include vomiting, lethargy, irritability, ataxia. Ataxia means loss of coordination and mental retardation in severe or untreated cases. So as nurses, we must recognize these signs, especially in newborns where these enzyme defects often first appear. Early diagnosis and dietary protein restriction can help manage these conditions. If left untreated, this condition can lead to coma and death due to ammonia toxicity. Now let us wrap up with our conclusion. The urea cycle is a liver's vital detoxification process, converting toxic ammonia into urea for safe excretion. By excreting with the TCA cycle, it's maintain both nitrogen balance and energy deficiency in the body. Precise regulation of this cycle ensures metabolic safety, and any disruption, whether due to liver disease or enzyme defects, can lead to fatal ammonia toxicity. As a nursing professionals, understanding this cycle helps us recognize symptoms of liver failure and hyperammonia, understanding why hyperprotein diets are restricted in the liver patients, and appreciate the importance of liver function tests like AST. And with that, I would like to conclude my presentation on the urea cycle. Thank you.